Welcome to Biology classes with Lakshmi Chandra. Having completed the definition of biotechnology, the tools of biotechnology, and I have also introduced to you what restriction enzymes are, today we'll focus on the action of restriction enzymes or how these restriction enzymes actually function. For that, let's focus on the steps. Okay, the whole action of the restriction enzyme I have put here under 4 steps. Let's see one by one. First, the restriction enzyme inspects the length of DNA sequence. For that, let me just write down a DNA sequence starting from 5 prime, and I'm just putting a sequence here GAA TTC, and then there are lots of bases here ending with 3 prime. Again, the second strand of DNA, we know they are anti-parallel and they are complementary. So here it will be C, A is going to pair with T, A is going to pair with T, T with A, again T with A, and C with G, and then there are other sequences here, and it is going to end with 5 prime because they are anti-parallel. Now this is the length of DNA, and into this DNA I have added the restriction enzyme EcoR1, and we are going to learn how restriction enzyme functions, taking the example of the restriction enzyme EcoR1. In the previous session, already it was taken like how this enzyme was named as EcoR1 and why it is named as restriction enzyme, what is the difference between restriction endonuclease, exonuclease, and such things. So it will be better if you go through those sessions first and then view this particular portion. The link regarding the previous sessions are put in the description. You may please see those description links so that it will be very useful for you. So EcoR1, how it is going to function. First, what this enzyme will do, it will inspect the length of DNA sequence. It will try to move from one end of the DNA slowly. It is moving towards the other end of DNA, reading the bases. That is, it is inspecting the DNA sequence. And then it finds a specific recognition sequence. It finds a sequence which for it is very favorable, and which when it finds that sequence it stops there. Okay, so second thing is it is going to find the specific sequence. That specific sequence for the restriction enzyme is called as the recognition sequence. It can recognize its sequence and it will understand, yes, I am going to make a cut within this sequence. So that sequence which a restriction enzyme understands that is its sequence where it has to go and cut, that particular sequence is called as the recognition sequence. So that recognition sequence is what I have written here, GAATTC. This is the recognition sequence of EcoR1. Now just look at this recognition sequence very carefully. From the 5 prime end, if I am just putting the recognition sequence, it is, you can read it as GAATTC. I am just taking the sequence alone. And again the other strand, if I write from the 5 prime end here, it is also written as GAATTC 3 prime. You see, the reading frame is same. The reading frame is same when read from one direction. When read from one direction. That one direction is either you start reading from 5 prime to 3 prime or you read from 3 prime to 5 prime in both strands. Reading frame is same. You start reading from 5' to 3', GAATTC, both strands. That means in the other strand also, start reading from here, it is 5', GAATTC. Again, you start from 3', CTTAAG. Again from 3', CTTAAG. So reading frame is same when you start reading from any one direction, either 5 to 3 or 3 to 5 in both the strands. Such sequences, such recognition sequences whose reading frame is same when read from one direction in both the strands, you call such sequences as a palindromic sequence. You call this as a palindrome, or you call it as a palindromic sequence. The reading frame is same when you read from any one direction, 5 to 3 here, and then you have to read in the other strand also from 5 to 3. Then you read the same thing. Reading frame is same. So that is called as a palindromic sequence. So ECO R1 is having a palindromic sequence as its recognition sequence. Okay, usually when this palindromic sequence is explained, we take the example of this particular word. I'll just write it here. What is this? Malayalam. You read from here, Malayalam. Language of Kerala. Okay, reading frame is same when you read from other direction. So this is example of palindrome. Same way, our ECO R1 is also having a recognition sequence. And what is the speciality of its recognition sequence? Its recognition sequence is a palindrome. So I hope you understood the meaning of a palindromic sequence. Okay, so first we are going to learning the steps of how restriction enzyme is functioning. First, it will inspect the length of the DNA sequence, and then next it finds its recognition sequence, the area where it has to go and cut. Then once it finds its recognition sequence, it binds to DNA. Okay, it binds to DNA. Then what happens? Then happens the actual process of cutting the strands. It cuts both the strands. It cuts both the strands. The two strands are cut. But not randomly. They are cut at specific points. The two strands are cut at specific points. Very specific points. And those points where they make the cut, those areas are called as the restriction sites. So you have the recognition sequence, the whole sequence which it recognizes. Then in that sequence it makes a cut at specific points in both the strands of DNA. Okay, and that region where it makes a specific cut is called as the restriction site. Restriction site. Let me just tell you what is the area here. Here the cut is made between G and A. So in this strand it is made between G and A. Here is the cut. In the other strand also the cut is at the same specific point between G and A. Here the cut is made. And here in both the strands. So we all know the bond which is actually binding the two strands of DNA, these two strands of DNA here, the bond between the bases, this is the hydrogen bond. But here which bond is cut? The bond within the strand is cut. So which is the bond within the strand? It is the phosphodiester bond. Phosphodiester bond. So restriction enzyme cuts the phosphodiester bond within the recognition sequence. That is how it functions. Okay, will it cut randomly? Never. First, it inspects the whole set of length of DNA. Next, it finds its recognition whole sequence. Then it binds to the DNA. And finally, it will cut the phosphor. It will break the phosphodiester bond at specific sites within the recognition sequence. So those specific sites are called as restriction sites. Okay. So having understood how this DNA recognition, this particular enzyme, restriction enzyme functions, in the next session I'll explain to you how a recombinant DNA is produced. Thank you for now.